Greetings, Pencrest biology students. It's Mr. Del Prado here, and today we're going to take a look at the first two reactions in cell respiration. So for those of you who may have forgotten or who just need a little refresher, I kind of moved the slides around a little bit for this, but I just wanted to start with this slide. So if you take a look at it, you can see that there's four different reactions in cell respiration. The big picture, if you get lost in all this, is that we're just taking glucose, which is the food that you eat, and we're going to turn it into ATP, okay? And that's going to take place over the course of four separate reactions inside your cells. Glycolysis, which occurs in the cytoplasm, transition reaction, which is going to occur in the mitochondria, citric acid cycle, and electron transport chain, okay? When we see these NADHs and the FADH2s, remember the real thing that we're going to do with that is we're going to rip those hydrogens off and we're going to pump them through ATP synthase to generate some ATP for us. Okay, so that's the big picture. What we're going to do now is take a look at step one, which is going to be glycolysis. Okay, so glycolysis is a whole series of enzymes that occurs right in the cytoplasm. We are not in the mitochondria. And if you want to know the big picture, and that's what we're going to try to keep our focus on, is we're going to take glucose, right? That ringed molecule kind of looks, I think it's a hexagon, right? Six-sided ring, excuse me, five-sided, and then it has the carbon on the outside. We're just going to take that glucose and we're going to cut it in half. When we cut the glucose in half, we call it pyruvate. So if you think about it, we're just going to take that ring and cut it in half. All right. If you're a bacteria, that's it. That's where you get your energy because you're actually going to make a couple ATPs from cutting that ring in half. But for eukaryotic cells, we're going to take those pyruvates and break them down even a little bit more. Mr. Dell, how could you possibly get ATP out of breaking glucose in half? Look, this is where things get a little bit intense. This is the process of glycolysis. Look at the enzymes. Hexokinase, phosphoglucose isomerase, phosphofructokinase. Okay? I am not asking that you know the steps for glycolysis. Okay? You do not need to know them. If you look at the bottom just for fun, you can see like there's half and there's half. There's the glucose that's actually cut in half for you into two pyruvates. What I really want you to know is what you get out of the chemical reaction. So you don't need to know all of these enzymes. What I'm asking that you know is this. When in glycolysis, we take glucose, we cut it in half, we get some pyruvate. We're going to get this NADH. Remember, we'll rip this hydrogen off a little bit later. Okay? And essentially what we're going to get is we're going to end up getting two ATPs, which is nice. It's two ATPs, but that's not enough to get the job done for a eukaryotic organism. So we get a little bit of ATP out of glycolysis, but the big thing that we're going to get are these pyruvates and this NADH, which we're going to then send to the next step or the next process within cell respiration. Remember I said it's four reactions. I just took you through reaction one. I'm going to take you through reaction two. Before we do that, I think the big picture you should know here. What happens during glycolysis? Glucose gets split in half to two pyruvates. Where does this happen? It happens in the cytoplasm of the cell. Okay? Reaction number two. Reaction number two is going to take place in the matrix of the mitochondria. So if you've forgotten what the matrix is, that's okay. The matrix is the inside, it's the gel inside the mitochondria. I think I said to all my classes yesterday, it looks like a river that goes to nowhere. Okay, so we're in the gel of the mitochondria. Okay, and in that process, what we're going to do is we're going to take each one of those pyruvates, and what we're going to do is we're going to use those pyruvates to drive a reaction where we make some NADH. In this process, okay, what's going to happen is we're going to make some NADH and then we're going to make this molecule. It's called acetyl coenzyme A. All right. And in this process, we're going to give off some carbon dioxide. And that carbon dioxide is actually going to leave the cell, go into your bloodstream, go into your lungs, and then you're going to exhale it. It's the waste product for cell respiration. Again, in the transition reaction inside the mitochondria, there's a whole bunch of enzymes that do this. We're not going to concern ourselves with all those enzymes. What we're going to do is we're going to look at the big picture. We're going to take the pyruvates that we made in glycolysis. We're going to rearrange them to get this molecule called acetyl coenzyme A. I have two acetyl groups here. I'm never going to ask you, the structure of those molecules. I will ask you this, though, the transition reaction. We're not making any ATP. We make some carbon dioxide that we breathe out, but then we're making this NADH. We're going to use this NADH later so that we can cut off this hydrogen and then generate a whole bunch of ATPs from it. Okay, so that's it. Those are the two reactions. Tomorrow, what we'll talk about in class is the citric acid cycle, which is another big cycle. We'll get some ATPs. We'll make some more NADHs as well. But we'll do that tomorrow, and then we'll finish with the electron transport system. The big picture here, so far through glycolysis and the transition reaction, we've made 2 ATPs. That's not enough. So I suspect you can figure this out during the last 2 reactions in cell respiration, especially the last one, the electron transport system. We're going to end up making the mother load of ATP. And I'm going to ask you specifically, like, how much ATP is made for each molecule of glucose. So far, through the first 2 reactions, we've made 2 ATPs, okay? Not enough. So what we'll do tomorrow is we'll take these acetyl coenzyme A, and we're going to make it go through another reaction in the mitochondria called the citric acid cycle, okay? And then the final reaction will be the electron transport system, also in the mitochondria, and that's where we'll end up making a whole bunch of ATP. Okay, so for those of you who are confused, lost, and scared, remember what we're going to look at are the big picture things. So these charts that are these plates that have just like the inputs and outputs, that's what I would end up focusing on. So big picture, okay, we've done glycolysis, transition reaction. In glycolysis, we made 2 ATPs, we made some NADH. The transition reaction, we didn't make any ATPs, but we made some NADH. And then you can see citric acid cycle and electron transport system both make NADH, FADH2, and they're going to make a whole lot more ATPs. Remember ATP, that's the gasoline that we use for our cells to give us energy. That energy is used to complete all sorts of processes in our bodies. But we will go over those last 2 stages of cell respiration in class. So I hope this made sense to you. I hope I didn't confuse you and I kept you looking at the big picture and not getting caught up in all the little reactions. As long as you know what goes out of for each reaction, I think we'll be okay. Okay, drop me an email if you have any questions, and I look forward to seeing you on the other side. Thanks for listening.